study the mechanism that protects such delicate organ that enables us to solve all these complexity problems. Okay, so here is the part one of today is to see the importance and the composition of the cerebrospinal fluid, and also how blood-brain barrier is formed and regulate our brain homeostasis. This is the learning objectives and the goals for this lecture. And the reason I want to, show this very detailed wording and very boring wording is to reinforce the concept that you really need to using these learning objectives to kind of assessing where you are, and have you gotten to the point that I would wish you to get out of from this lecture. So there's some useful tips for using those learning objectives. Is that you probably can tell that all these verbs, this does not show the laser point. Okay, so the diagram lists to explain, and all these words can actually help you. That's create a question for you. So if you are able to actually do all these things we listed here, you're in a good shape. Okay. Since the brain is so important and it has to be very well protected, your body has three different ways to protect it listed here. Starting from the cranial, the bone of your brain. Those cranial bones we talked about in lecture previously and also in lab, so I'm not going to spend time to do it. And underneath these cranial bones, you can find the three meninges layers. Okay, so we're going to talk more about the meninge layers in a couple of next slides. And also, there's a very important fluid in your brain, finding your brain. Most precisely is surrounding and inside of your brain to help to absorbing any shock and mechanical damage to your brain. So here is a diagram showing you three layers of a meninge. We peel them one by one. So the first one, as we show on the left, is called a dural matter. Dural matter is a layer very hard. Precisely, it actually have two layers there. So you can see that's a very hard fibrous layer. And this, in the middle is a arachnoid. Arachnoid is lying underneath the dural matter. Okay, so it's the middle layer in these three meninges. And finally, if you peel the arachnoid membrane very carefully, you can see on the last picture, is the right side of the brain is showing your peel matter. Okay, so you can see the differences. What is the difference between the arachnoid and the peel matter? Just by looking at it. What's that? One is like a film. How about the other one? The one is a film that covering the whole entire brain, does not penetrate and follow the folding of your brain. The other one does, right? So starting from the first one, the dural matter. Dural matter is called a hard mother. The name is suggesting it's very, very hard. The tissue is very hard. It has two layers, two fibrous layers here. And in between of them, you can find a very slender gap, creating a space for the veins and the arteries and to go in. Okay, so they have through tissues there are also blood vessels finding in between of these two layers of the dural matter. Underneath this is our arachnoid membrane. It's epithelial membrane, so it's not as hard as the previous one we just talked about. But the arachnoid membrane has this very special case here. Is right underneath the arachnoid membrane is a space. Okay, this space we call the subarachnoid space. This space is very important because it is where the cerebrospinal fluid is surrounding your brain. Okay, you can find the cerebrospinal fluid underneath, inside of this subarachnoid space. So underneath the arachnoid membrane. Okay, as we talked before, this membrane does not follow the fold of your brain, unlike the pure matter. Pure matter is the last one, the most inner layer. So this one does follow the brain's underlying fold and accompanies the branches of the vessels that neutralize the brain, just the blood supplies to the brain that comes with it coming in. Okay, we talked about how important the subarachnoid space is because of this create a place for the cerebrospinal fluid surrounding the brain. And to understand the cerebrospinal fluid, we have to talk also about the ventricles, brain ventricles. So here, both from the lateral view and the anterior view, we show the blue colored region is your ventricles. There are 4 ventricles in your brain. So when we talk about ventricles, these are the space that are actually filled with fluid. In this case, the cerebrospinal fluid. So we see that there's 2 lateral ones, a paired one, lying enclosed by your cerebral cortex. Okay, so the cerebrum is these 2 lateral ones. We call the lateral ventricles. Sometimes people call the first and the second ventricles. Both of these ventricles are connected to the third ventricles through the interventricular foramen. Okay, here. And once they connected to the third ventricles, it keeps going. The third ventricles, as we see in this anterior view, is very, very thin. Okay, oftentimes people call it slit-like. So it's a very thin structure, the third ventricles, and it follows and it continues to the fourth ventricles. The structure or the channel that connecting the third ventricle and the fourth ventricle is called cerebral aqueduct. Okay, so here is basically the very important several structures: the lateral ventricles, third ventricles, and fourth ventricles. And we also have the connections, which is the interventricular foramen and the cerebral aqueduct. Cerebrospinal fluid, as we see, is surrounding your brain and also finding inside of your ventricles. And the reason it's important mechanically is because of the fluid all around your brain make your brain actually weight a lot less. Could you imagine if your brain does not have a cerebrospinal fluid, how heavy it weighs and how much damage just every time you take a walk and your brain will bounce into the bones that's connect to it, the membrane and then bones. So this cerebrospinal fluid does creating practically lifted the brain inside of your cranial, inside of the cranial bones, and helps to decrease some of the effective weight. Okay, so it providing the cushion and protection. Another important role that the cerebrospinal fluid play that the other 2 mechanism does not is actually maintain the chemical stability, which means the cerebrospinal fluid is constantly generated. So your brain every day will renew this cerebrospinal fluid about 4 times. And so when you're renewing, if there's any toxin, there's any chemicals that you want to get rid of, that will actually washed out by those cerebrospinal fluid because it's connected to the interstitial fluid. So those are the important point about the cerebrospinal fluid. And as we can see here, just before we move on, and this blow up here, the little picture here showing you the subarachnoid space. One of the reasons I didn't say what the arachnoid means. Arachnoid means like the spider webs, the network. So you can see there's a lot of like kind of a network looking thing that was the cells and the fibers that are coming from the arachnoid membrane and connecting to the pure matter. So that's why they got the name. The circulation of the cerebrospinal fluid is actually probably a little bit of review for you guys, because I heard you talked about how cerebrospinal fluid is generated. What kind of glial cells responsible for generating cerebrospinal fluid? Very good. Ependymal cells, Okay, where you find those Panama cells? Okay, central nervous systems precisely is the choroid plexus in the ventricles. So you have 4 ventricles. There are 3 of them actually have those choroid plexus, and 2 of them are in the floor of your lateral ventricles. Okay, so the other part is the roof of the 3rd ventricle. So you also have those choroid plexus. So together, these regions are constantly generating cerebrospinal fluid, and after they generated, they continued, because we talked about the structure, it's all continuous. So they're going to go into the 3rd ventricles, follows to the 4th ventricles, and then leave the 4th ventricles through the pair of lateral apertures and the medium aperture. And by the time they're leaving the ventricles, they are going to be able to surrounding the whole entire brain in the space we have already said many times, subarachnoid space. Okay. So after this, we need to take away this cerebrospinal fluid. The generation of the cerebrospinal fluid is not is pretty constant. Okay, it's not regulated by the pressure inside of your brain, but the absorption is affected by that. So if you have increased pressure inside of the brain, and you will have more fluid leaving, and where does they go? Is these places, little inserts here, showing you the arachnoid villi, and so they will be drained into the durovenous sinuses, which is between of these 2 layers of the dural matter. Okay. So we're going to use the first time the clickers in this class. Have you used the clickers before? Here is a couple very easy ones, and turn on, you just they're on and off button. And this classroom is the channel A and C. So you need to hold on to your on and off button for maybe 2 or 3 seconds until the light flashes. Okay, do you get it? And then press N and C. So is everybody okay with this part? Okay, let's try it. All right, another thing I would like to say is, in my previous years, I always do this. If you come to class, okay, besides the quiz, that's one way you can earn your extra credit. If you come to class and participated in these activities, for example, the clickers, and I'm going to go home and see who's here and who answered correctly. And I want to say, if you answer 1 question correct in each class period, you get 2 points. Okay, but maximally you can get 6 points in total. We don't want to really inflate the exam, but just trying to inflate your grades, but just trying to get everybody participate. Okay, so if you when you have a dog, always talk to your neighbors. That will be helping you to getting the question right. But if you, basically I have 6 lectures before the 3rd exam, so if you've been here 3 times and answer 1 question right, in each lecture, you are all set with that 6 points. Okay, so got a good number of people answered already. Okay, very good. So, diffusion. Why people think it's not an active transporter? Why not an active transporter? If it's an active transporter, you won't be able to see they have a very similar ion concentration. Why is not osmosis? Osmosis is similarly. Osmosis are only able to, that means you're able to move the water freely. So in other words, the osmolarity will be similar between these compartments, but the concentration of the ions would probably be different. It depends on what kind of channels are existing. Those should be covered before. So another note just by this question is your interstitial fluid and your cerebrospinal fluid is actually have identical ion concentrations. So they can freely communicate with each other. It's unlike the next topic we're going to talk about, which is blood. Okay, so the blood is important because the neuron tissues is very delicate and they are very prone to any hypoxia, ischemia, and so you need to have a very, very rich blood supplies to your brain. This part is not covered in your book, but it will be in your exam. This is many of those things if I mention it in class, and this is a big point in class, we talked about it, it will be your exam. But if we didn't talk about it in class, and you don't need to worry about it. But always reading books is a good thing, even though some of the topics we don't get to discuss in details. Okay, so here, the blood supplies to your brain is entirely supplied by two sets of arteries. And one is called the internal carotid artery, which giving rise from the common carotid artery. And the other one is called a vertebral artery, which comes from the subclavian artery here. Okay, so these two sets of arteries are giving the whole entire brain blood supply, and the blood will drain through these internal jugular veins. We're going to take another close look at how these arteries supplies the blood to your brain. So here, internal carotid artery were further divided into two main branches. One is called anterior cerebral artery, and the other one is called middle cerebral artery. And the vertebral artery on the other side is coming from the behind, okay, from the posterior side of your brain. And at first it becomes a basilar artery. So these two arteries actually merge and become basilar artery. And then it will further divided and becomes called a posterior cerebral artery. Okay, so posterior cerebral artery giving blood supplies to the posterior side of your brain, and then anterior and the middle cerebral artery providing blood supplies to the frontal part of the brain, middle part of the brain. This diagram is showing you a close look about how these arteries supply the blood to your brain. Again, anterior cerebral artery is coming from the middle of your brain. Middle cerebral artery coming from the lateral side. And the posterior coming from the back. Okay, so here is actually one of the interesting part is that even though all arteries are prone to clotting, and there's one set of arteries that's particularly prone to clotting, which is the middle cerebral artery. So this is why many of these patient has a stroke. They're actually losing functions in speech and maybe movement. That's because of a lot of these brain areas that were contributing to these functions are located in this region. So left brain and the superficial part of the cerebral cortex. So maybe this is an overstatement because of a lot of other arteries that can clot and do not contributing to the symptoms, right? So these are symptoms we can quickly noticing and identify, and then we know we had a stroke. But sometimes you can have a clotting inside of a brain without even noticing it. So, but people still many times, if they say any artery that's prone to clotting, they will mention that it's a middle cerebral artery. One of the interesting factors of your brain, the artery supplies, is actually if you're tracing the cerebral, the vertebral artery, and then it becomes the basilar artery. If you're looking at the right picture, it is more obvious. And it's actually connected with the arteries that are coming from the anterior and the middle, and creating a structure like a ring shape at the base of your brain. Okay, so now you can see that this structure what is the structure good for, if anything? Do you think is there any advantage to have a structure that has a ring shape at the bottom of your brain that is connecting all the blood vessels? Yes? increasing the efficiency of the blood flow, that might be, but that's not the answer I'm looking for. Equal distribution. So this is a very close equal distribution. Anything else? Not exactly what I was looking for, but this is a very good. I think actually this is in the same line of what I'm looking for exactly. Yes? Circulation, providing what kind of a circulation? What I really do is, what I'm looking for is actually the backup, safety check. So if you have a clottage in one area, and if you have this ring shape, it may not, the tissue that is supposed to be deprived of the blood actually be able to have the other vessels supplying the blood to that region. Okay, so it cannot be really downstream, but if the clotting happening majorly in some areas that could be compensated by the other blood vessels, and this circle of Willi does help to prevent the major damage to your brain. So this is an example we call a collateral circulation. What is a collateral circulation mean? It's basically one area of the tissue has blood supplies not just coming from one blood vessel, one channel, but from many other channels. So this is called a collateral circulation. So another question for you is, what do you think the composition of the brain interstitial fluid compared to plasma? Is it similar or different? This is the, how many of you have iClicker 1? If you have iClicker 1, every time you turn off your iClicker, you need to, when you turn back on, you need to change the channel again. So it's always reset to AA, so you have to reset that one. You can probably tell I'm trying to give out the free points. So please participate.
Mm-hmm. I'm gonna do so much better in the next 16. How'd you do in the previous one?
Okay. I'm going to stop here. Oh, sorry. Okay. That's okay. See, here's the misconception. We do need to review those and correct them. So what do you think about so here, have you heard about the blood-brain barrier? You heard about it. What do you think the blood-brain barrier will do? If you say you haven't heard about it, I'm going to tell Dr. Jangunas he could probably cry at home for a couple of days. We talked about astrocytes, right? That was when he mentioning about blood-brain barrier. Maybe you have some kind of contribution to it, right? So that blood-brain barrier is the mechanism or structure that preventing a free communications between plasma and your brain fluid. Okay, so because of that, you can kind of think why we need that. Not everything in your system, you want them to reach to your brain. Okay, so brain is so delicate, you want to protect it as much as you can. So this is why we don't see a close or similar ion compensation and a protein compensation between these two compartments. Okay, so the correct answer is different. B. Okay, so here is a couple of things that's showing you how the interstitial fluid and also the cerebrospinal fluid different from your plasma. Here's some main differences that I picked out for these two different compartments. So first of all, your CSF has a much higher partial pressure of CO2. And because of that, you have a lower pH value. The pH value in your brain is lower than your blood. And you have a very cerebrospinal fluid has a very low protein content. So because of that, resulting in very low buffering capacity, which means that ion, for example, the hydrogen changes, your brain is more prone to those changes because the protein sometimes can buffer a lot of those. Okay, so they have a low buffering capacity in your brain, in those cerebrospinal fluid or the interstitial fluid. Cerebrospinal fluid has a lower glucose concentration. Okay, your brain really needs, we're going to see that in a second, your brain does have a very high demand for oxygen, for glucose. So why you think the cerebrospinal fluid will have a lower glucose concentration? Okay, this one I will give out. Okay. The glucose will be actively transported inside of your brain. There are transporters doing that. But if my question is, it's a kind of contradictory. It's a kind of a counterintuitive, because if your brain needs glucose, why it has a low concentration of glucose compared to anywhere else? This is because your brain cells has a very high affinity glucose transporters that will actively transporting the glucose inside of the cells and use it for energy. Okay, so that's why you can see CSF has a lower glucose concentration. They have a high chloride concentration, low cholesterol content. Okay, so they are different. This is a review, and we talked about how brain has been, how much brain blood reaching to your brain. Quick number, 15, right? 15% of the blood pumping constantly goes to your brain, and that means 15% of oxygen that you inhale goes to your brain as well. And oxygen can, as a CO2, they both can pass freely through the blood-brain barrier. Glucose, our brain uses about half of the glucose that in your body, and because of the glucose, your brain is very picky. As the next semester we're going to learn, it does not use many other new kind of molecules like free fatty acid, as they cannot use those as their fuel. So they have a high demand for glucose. The glucose, about half of them used there in your brain, and there's membrane transporters that move glucose from the plasma to the brain and from the interstitial fluid to the cell. So these transporters has very high affinity for glucose. And if there's a glycemia, if the hypoglycemia happens, which means I have a low glucose level inside of your system, if your plasma glucose level drops, you're going to have unconsciousness, and there will be confusion, dizziness, and eventually cell death and the death of the body. And this diagram, I was fascinated by it because it's an elephant, it's an African elephant fish, and about 60% of the oxygen and the blood supplies goes to the brain. Enormous, big brain. Compared, just the idea compared to a human and the other species. All right, one of the big the big topic of our this class is to talk about the blood-brain barrier. Okay, so blood-brain barrier, what is forming the blood-brain barrier? This is, I heard it is a review, so I threw this question to the class. Astrocytes. I heard astrocytes. Astrocytes is actually a very important portion of in the blood-brain barrier. However, not every part of your brain have astrocytes present with this. Even though they have blood-brain barrier, some of the area do not have astrocytes. This is just a point of interest, but it's very astrocytes is historically people think is important for blood-brain barrier, and I will go to talk a little bit about how it works. Anything else? Anything else? blood-brain barrier. What do you think would make your brain is so tightly controlled? What is the first part that your blood will leaving the vessels? Any answer is fine. I feel like I'm giving out the free points and you guys just, you say, oh, I'm going to actively participate, but you don't really want. Okay, anything else? Just looking at the picture, what are we showing here in that picture? Capillaries, and what is the cells that's enclosed it? Endothelium cells, right? So endothelium cells found in this region is very, very, very special. They have formed a very complex and tight junctions. So it is actually 50 to 100 times tighter than any other regions with the endothelium cells. Okay, so as we can see here, the pointer doesn't work very well, but this is the cell body of your endothelium cells. As we said, this enclosed capillary lumen, and you can find the very, very the tight junctions in here. I don't know if you can see it. Since the tight junction is so tight in those endothelial cells in your brain, and they limit basically they limit most of the paracellular transporters. Okay, so they are also ensheathed. Is this a microphone works for you guys? Okay, I find it's a little disturbing. But, so you can also find many astrocytes that is surrounding these endothelium cells. Okay, so we call it the ensheathed by astrocytes foot process. So together they regulate the composition and the volume of the brain interstitial fluid. As this is a more close look about how these astrocytes run the feed process and enclose here. Functions of the blood-brain barrier is listed here. They control, obviously, they control the molecular traffic, keep out the toxin, and it is one thing we will very much want it. If you apply a drug, if some of the drug, for example, this treating the asthma or treating the allergy in the spring, and some of the drug will give you kind of dizziness and nauseous. Some of the drug does not. The reason being is some of them are able to cross the blood-brain barrier and some of them do not. And the reason they will be able to cross, oftentimes either they have transporters or they are lipid soluble, they can diffuse through the membrane. Okay, so they keep out the toxin. They maintain the iron homeostasis inside of your brain. This is extremely important because your brain needs to do a lot of firing, a lot of action potentials, and you have learned how important to maintain those potassium, low extracellularly, high intracellularly, sodium, vice versa. So those are all important. And if you don't have a very good way to protect your homeostasis in the brain, for example, you drink a bunch of V8 and you eat a bunch of banana, and so maybe that will be changing your the compensation of your brain. We don't want that. Any of the chance to get changing those potassium and sodium concentration. They have very low protein environment, limited proliferation. The separating central nervous system and peripheral nervous systems, which is important because of this allows, as we can see later on, there has a lot of neurotransmitters that is diffused throughout of your brain, does not need a cell-to-cell communication. Okay, so that allows you to have one kind of neurons and project the many other regions, and also affecting more of these other neuron group. That is why. Okay, so you have a very tightly controlled environment to allow them to do that. And then last but not least, immune surveillance with minimal inflammation and cell damage. Okay, so you don't want to let T cells, B cells come in and wipe out your nervous system if there's any suspicions going on. So the ones with astrocytes are listed here, these tight junctions that as we just talked about. So there's a growing evidence suggesting that the blood-brain barrier is oftentimes achieved in a unit. We call it a neurovascular unit, NVU. Okay. So this unit, including vessels in that region, including the neurons. So we have a lot of neurons giving input to the vasculatures. And so, for example, later on we'll learn in this semester is how autonomic nervous system can change the diameters of your vessels. And there's a glial cells that play also in a role in this this picture. So there's two glial cells that we particularly will mention. Is one is a microglia, and one is this astrocytes. Okay. So as I mentioned before, astrocytes, a lot of people has been, very strongly believed that it's very important in the blood-brain barrier. And later, we have a very heated debate on how astrocytes is contributing to it. And it turns out, if you culture astrocytes into those endothelium cells, okay, so if they originally, in the culture, if they originally do not have astrocytes, and now you culture them, the astrocytes into it, they make the tight junction much tighter. Okay, so those are, they have a induction role in terms of forming those tight junction. And the microglia, on the other hand, also plays some permissive role in getting the environment better for the tight junctions to form. So here we have astrocytes and microglia. We also going to listed in this diagram kind of a greenish looking cells surrounding the endothelium cells. This is the parasite. Parasites is one of the cells that has very smooth cell-like, so it has a contractivity. So those cells have surrounding those vessels and believed to controlling the diameter of those vessels and contracting these the vessels we're interested here. So altogether, they regulate the regional blood flow as well, controlling the ion and the fluid transport between your blood and your interstitial fluid. So if you're interested, here's another paper that I have a review on this topic. Okay. So we mentioned that there are some gases that can freely diffuse, like the O2 and the CO2. How about ion? We need ions that we need transporters, we need channels that will be able to move the ions in and out of the cells, in and out of the plasma. So here, I have to show you another terminology that be careful with. Is just by convention. Whenever we talk about ions that moves in and out of the plasma into the cerebrospinal fluid or into the interstitial fluid, we're talking about the secretion and absorption. Okay, so secretion always means both terms always refer to the blood. If you have a secretion refer to the blood, what do you think the movement will be? Going from the blood to the interstitial fluid. Is that acceptable? Okay. So whenever we talk about secretion, it's always moving from the blood to the interstitial fluid. And whenever we talk about absorption, it's always coming from the interstitial fluid and then come into the blood. So here, that helps us to understand the title. Why they refer to blood, I don't know, but maybe because of those pioneers who studied this region. But this is a convention now. So in the exams, if I say secretion or absorption, don't feel that, don't argue about the points. Okay, so secretion here is we have a secretion of a sodium chloride and water into the brain. Those blood-brain barrier were constantly doing that. Does that make sense? Okay, what kind of ion concentration you should find in the interstitial fluid for the sodium, high or low? Sodium. High, very good. You want to find a high, so secretion makes sense. Absorption of the potassium into the blood. Does that make sense to you? Right, so you wanted to have a very low extracellular potassium level surrounding your neuron. So you want to constantly removing them, absorbing them back into the blood. So that is why we say it is important providing the optimal signaling environment for the neuron to fire in action potential. So there's a lot of disease associated with the disruption of the blood-brain barrier. And I only listed here for your interest. I'm not going to quiz you what kind of a disease involved in the blood-brain barrier disruption. And if you can argue almost all neurological pathologies have some aspect that will changing the blood-brain barrier. So here the labeled red are those who has issues with the blood vessels. The greens associated with infectious agencies like bacteria, virus. And the blue labeled here are the neurological disease like epilepsy and depression, schizophrenia. And the black labeled are related with aging. So as we grew older, and those happens, and these can also disrupt your cerebrospinal blood-brain barrier. Okay. So let me just quickly take a look. What time is it? 9:05. Very quick. I'm going to quickly go over this. We're right on time. This is a promise. This is probably the lecture that's the least interactive. We're going to get it more interactive in the future. So stroke is just using one of the examples showing you how disrupted blood-brain barrier can, what kind of the stroke can, what damage the stroke can do to your blood-brain barrier, and how that will influence your neurological functions. Okay, so stroke has two different ways. You can have a clotting, you can have a basically blockage. You also can have a bleeding. So ischemic stroke was referred to this blockage. So it could happening due to the cardiac arrest, or it could happen with a clotting inside of your brain in one of the cerebral arteries. And the other one is the hemorrhagic stroke. This is when you have a burst out of your vessels, and you can see that that's clearly going to disrupt your blood-brain barrier. And we want to talk a little bit about the not so obvious clottage, what is going to happen to your blood-brain barrier. Okay. So here is the figure having several players here. We have a neuron, we have a blood, we have neurons, we have endothelial cells, we have free radicals. So what happens is when you have a blockage in any of these vessels, the downstream tissues is going to suffer. Okay, so they're going to deprive of the oxygen and reaching to the hypoxia. Your neuron is so prone to those changing of environment that they will die and send out the free radicals. You normally have free radicals in your body all the time, but when this neuron dies, those overwhelmingly secretion will causing these endothelium cells to die. When the endothelium cells are damaged, you can imagine they will causing the blood-brain barrier will be compromised. So here are some early events. The early event is a rapid development of the hypoxia, as just we talked about. And then we're going to have also the central release of the vasopressin. Have you heard about the vasopressin before? Okay, this is one neurotransmitter or hormone, whatever you call it, released by the hypothalamus. And then they reach to the regional, so systemically, what it does is actually trying to retain water. They noticed that what is the signal received is the body needs to retain water. So what they do is they're going to constrict your blood vessels and to increase the absorption of the blood, retain the water. So when this happens, together with the endothelium, transendothelium secretion of sodium chloride, as we just talked about, what you can envision happening is brain edema. Okay? When the brain cells start swelling and the tissue starts swelling, what's going to happen is they're going to press against these vessels, causing more of the blockage. So the region, the ischemic region, the zone started to spreading. Okay. So afterward, later event is after 4 to 6 hours, and you can imagine the free radicals start walking, the endothelium cells start to damaging, and you have the breakdown of the blood-brain barrier after normally 4 to 6 hours, and causing the more systemic neural injury. Okay, so we know by now, this is a summary, blood-brain barrier, regulator of the brain interstitial fluid volume and the composition. And if you alter it during this blockage stroke, and that leads to the cerebral edema to form, and the edema is the major contributing factors for any of the morbidity and the mortality of stroke. Okay, so here I have this is assignment for today. Remember, I want you to review what we learned. So here's this assignment is use the rest of the class time or any time you can find to construct a concept map with partner using the following terms. Blood-brain barrier, cerebrospinal fluid, blood interstitial fluid, ion concentration of brain, stroke, edema. You may add any terms you want. For example, sodium, chloride, potassium, and how this is affecting the neuron function. Are you going to see hypopolarization or depolarization? And those are the things that you may want